Okay, so in our notes, when we finished up last, I kind of was rushing through the last slide, so I'm just going to go back and review that a little bit because it was towards the end. So just to remind you, what we're talking about are tools used in biology and other sciences as well. And so the last place where we left off was on how we separate and how we analyze different mixtures and different substances. And there's a few different tools that we use. We will use one of them, kind of simulate one of them, and one we won't use at all. But this is a way to separate out different molecules. The first one is the one we will do in class as part of one of our labs later on in the year, and it's called chromatography. Okay? Chromatography is separating out, usually a liquid, and separating the various solutes that are dissolved. Okay? Yeah, we did this. So it separates the parts of a mixture by several factors, okay? By their density, by how much they adhere to the chromatography paper, by their solubility. Basically, you have like a special type of paper, and the bottom, you sort of hang it vertically. I'll show you the picture in a minute. And then the very bottom is in a solution. And as it absorbs that solution, the solution starts to move up the chromatography paper, but anything that's dissolved starts to separate out. Okay? And it forms different layers. And then you can look at those bands that are formed, and it tells you a little bit about those various solutes. I'll show you a picture in a minute. Electrophoresis works in sort of a similar concept, although you set it up differently. We won't. This is one that we kind of model and we talk about how it works, but we don't actually do it. I think I gave away my actual electrophoresis equipment to Mr. Rogan at the high school. He teaches a biotechnology class. But basically, electrophoresis you can use to separate very small molecules. Generally, we use this for DNA or proteins. And you have a gel. You put samples of DNA or your protein in the gel. It's kind of like Jell-O. And then all right, so out, quiet in the hallway, rushing down the hall.
Really? Bumps?
All right, go ahead, guys. No talking in the hallway. 0. Left. We got important stuff to do.
Why is it longer?
All right, go.
Hey, I'm pretty sure Mr. James is on our street. He does? I'm pretty sure he does. Where does he live? Oh, you know where Matt Fisher lives? Yeah. I'm pretty sure he lives right across from Matt Fisher.
All right, let's get going. So, as I was explaining, electrophoresis, you have a gel. You put DNA samples in various parts of the gel. You then add electricity, and because DNA is a charged molecule, the DNA molecules start to move through this gel. And the distance that they move depends on the size of the DNA fragments. And it creates a series of bands that you can then look at to analyze. And the last one is spectrometry, which is when you can take an unknown sample of something and basically figure out which elements it's made out of. How much of it is nitrogen, how much is phosphorus, how much is carbon, and so forth. Okay? And really you do that by sort of burning it and looking at the light that comes from it, because different elements produce different patterns of light when they're burned. So this is chromatography. So you put this is the part that's sort of hanging in a solvent, and as the solution rises up the paper, it separates. This would be like different markers, water-soluble markers that they separate out, because markers are made of different pigments put in there, and the pigments separate out. It's also often done in, this is a leaf extract. So if you smash up a leaf and dissolve the material in a solvent, and then you use chromatography, that the liquid will travel up the chromatography paper, but there are different pigments that make up the pigments in a leaf that give it its color. There's different types of chlorophyll, there's other pigments, okay? And so they all sort of separate out, and that's what you're seeing here. You're able to separate out those pigments so you can analyze them. This is an electrophoresis equipment, whereas, the gel is in this rectangle here. You can see the wires for the electrical signal, and the DNA migrates. Then when you stain it, it forms a pattern of bands that you can gather some information from. And this is a mass spectrometer, basically a computer tool that you use.
Students, good morning. Just as a
to
reminder,
identify
during any
substances.
type of drill, the expectation is you will remain silent, exiting the building and coming back into the building. No talking until you get back into your classrooms, and then you go from there. Thank you.
All right, our last type of technology is imaging. And you're probably familiar with some of this. That we use various tools for imaging. And one of those is, you may have heard of a CAT scan before. Heard of that? Anyone like in that Grey's Anatomy and watch a billion episodes of it? No? Yeah, it was my daughter. For a long time, it's all she did. But anyway, a CAT scan stands for computerized axial tomography, sometimes called a CT scan as well. And basically you have X-rays, a rotating beam of X-rays that produces an image. But it gives more information than just a typical X-ray. Okay? It allows you to see sort of differences. Because of the difference between bone density and soft tissue density, they absorb X-rays at different rates, which allows you to see the differences and image that tissue. There's nuclear magnetic resonance in different types of NMR, and you're probably familiar with one of those, which is the MRI. Has anyone ever had an MRI done? If you ever had like a joint injury or something, they may do an MRI, and an MRI creates an image. It's often used for soft tissue injuries. Okay? Basically, you go in this big tube often that has very, very strong magnets in it. And these magnets cause the nuclei of individual elements to sort of orient themselves in a certain way. Okay? And then when these radio waves are turned off, the nuclei return to their original orientation, and when they do that, they emit a signal. And the computer sort of detectors pick up these signals, and it's used to build an image. It's often used too, it doesn't use any ionizing radiation, any harmful radiation, you know, the x-rays. You go get an x-ray on your tooth at the dentist, they put that lead blanket over you so that other parts of your body don't absorb these x-rays, because x-rays can be harmful if you're exposed many, many times. Okay? The MRI doesn't use any of those harmful radiation. And the other type is a sonogram, sonography, ultrasound, they mean the same thing, in which sound waves are used to create an image of underlying soft tissue. And you're probably familiar with a sonogram from when a woman's pregnant. She may have a sonogram of her fetus that's growing inside of her. But you also may get a sonogram or ultrasound to look at, you know, your knee joint, or to see if your spleen is inflamed, or you can image soft tissue using ultrasound. Basically, it works like sonar in a sub or that a bat uses by sending out sound waves. Okay? Now they're beyond human range of hearing, but they are sound waves, and then listening for them to come back. And by interpreting those returning sounds, an image is formed. Okay? So this is a CT machine, okay, to do a CAT scan. Okay, that's a CT scan of a person's head, and you can see, you know, their brain, you can see their sinus cavities, you can see their eyes, and it sort of forms this layered image where you can go through the various layers to look at different parts of that person. Okay, here's an MRI again of a person. It's a person's head looking from the side, and you can see the brain tissue and so forth. And then we have sonograms. This is a fetal sonogram. You can see the fetus inside of the womb. All right, so any questions about any of these lab biology tools that we've talked about? So your quiz on Thursday is going to cover this note packet. You have to review this. We'll talk a little bit more about it tomorrow. We're going to have a quiz on these lab tools. Nicolette?
Do you have a stapler?
I do. In a minute.
And a stapler?
Yep, 1 minute.